I'd like to invite our panelists up to the front for a panel discussion. And if you have a question for your panelists, please raise your hand. We can get a mic over to you. So.
Thanks for some great presentations. My question is for Phil. You mentioned that you're only reporting the variants that you know the function of. And most of the variants that we know the function of are not defined by studies that include diverse populations. And since diversity is one of the goals of the All of Us program, I wanted to know how you guys are thinking about that and the discovery of the novel alleles in diverse populations.
Yeah, it's a really great point. I want to make the distinction between returning and having the data. It's all cap CLIA. We can go back and return anything we can, but we have to have a claim and able to advance it. And the perception was that providing uncertain results makes things better, at worst, not better. So I would encourage the group and to use all of us to be able to make those associations. And then once those associations are made, our goals are to, just like adding new variants and alleles, to be able to rapidly bring them into the pipeline. But without the data supporting the associations, we can't make the assertion that we can return.
Hi, yeah, Peter Shawmerk. Question for you on the report there. Do you actually give the phenotype in the report? So I saw you had the drugs, not what to do, and the gene, but was the actual...
It is, yeah, I didn't show that page of it.
Thanks.
If you go to the website for NIH for All of Us, there is a sort of a mock thing that is the better example. We prefer to direct folks there than to do anything more detailed than screenshots. But yes, it has gene, genotype or diplotype, predicted phenotype, and then as you go lower in the report, the associations I saw you are listed. So it is a static report. If those things change, we have to go through some change management to be able to update them. Yes.
Not to predominate your time, Phil, but if maybe a comment from both you and Christine. A lot of people are getting the All of Us study performed. There's a number of participants. Number one, does that preclude them from doing a broad panel test if they are interested in doing so? Because so if they want to do a follow-up test with a larger lab, and then I'll wait a second for a response on that, and then Christine, a question.
Yeah, I wouldn't think why would that do that? So I mean, you could go through a direct-to-consumer test and get something back, and again, it's not a valid clinical test, so it won't affect reimbursements. I like the breadth of testing for a research return to be wide because it's the best predictor of whether you're going to find something clinically if it was a good research-based test. So again, we're not using it for clinical care, but we've yet to have one at our institution that didn't match. So if you're having a conversation and you're trying to talk about whether a test that costs $300 or $400 is something the patient wants to pay for, you know, it helps us direct testing maybe, but I don't think it precludes broader testing. If anything, it shows the value of it because, you know, the more you have, the more you see you can justify testing in the future.
I have a microphone, so I get to ask a question too.
Completely
what percentage of people, do you know, are getting clinical testing, or are you tracking that in any way?
We are tracking it. I don't have it today, but the tracking is limited to what they say they might do. There's no formal way of tracking that currently, but it's been discussed.
Hi, this is for y'all, I suppose. In many of your presentations, you're using language that mirrors the FDA's new stance on laboratory-developed tests and calling them in vitro diagnostics. And with that proposed rollout of that and over the next 4 years, what do you see the impact being on PGX as far as the ability to get the actual bench work done for this testing? Thanks.
What I have heard through, before the rules actually passed, there was an earlier presentation, and the implication during that was that a test like a pharmacogenetic test or a test that is polygenic risk scores is not the same as the LDT to IDE, and that the enforcement of that will actually begin before. I haven't heard any, we obviously in the community, we're all listening and wondering who's going to get tapped on the shoulder next. But my expectation is that they will begin enforcing fairly soon.
I don't have much to add other than the first milestone was it being determined as significant risk. So if the regulatory enforcement aligns with the degree of risk, then this is just an opinion, but I expect pharmacogenomics would be in that category.
I didn't read all 8,000, no, it was 188 pages, I think. But Bruce Quinn had a summary of it, and there was a call out for New York CLEP accreditation. So I think it's always a good idea, and I know it doesn't make any sense if you're a regional lab in, XYZ to go through New York, but it's always been a pretty good standard of approval for a lab anyway. It's probably not a bad idea to think about getting New York accreditation for the lab, or working with a lab if you're partnering that has that in place.
I'm curious, Phil, if a lab wanted to go through the same process you did and extend it out to, the extent of a commercial test, what do you think that would cost?
Oh, geez. A lot. I mean, 19 months, just number of people I was in close connection with the genome centers was way more than the numbers you said earlier. It was 30 to 40 people, and that wasn't even including the regulatory groups or the times or the back and forth. So I think it's significant effort. My concern not only with cost is the speed. It's you get through 19 months and you're already outdated. So unless there's an acceleration of the subsequent one or a clear path, it becomes very difficult, I think, for any type of lab community to be responsive to clinical need.
So forgive the onslaught of the all of us questions, but what's the relative percentage of the participants that want their genetic results back? And following up on that, are you able to follow them post-testing to have a sense of whether there are a differential number of adverse drug events in those that want the results versus those that don't?
Yeah, and that's a layered question. Everyone's interested in doing that, to be very, very clear. We have the numbers that we're interested from the very beginning. I realize that some folks, like myself included, entered the study like in 2018, 2019, and there's a long time frame. So I would be cautious understanding whether that number is stable based on time or reinforcement. There's attrition in any study. I don't have the percentages right here, but a significant number of people are coming. When I last checked, it was well over 100,000 people who had results returned. So it's not 225 dropping to nothing. It is a significant portion are coming back even with time. The ability to follow moving forward, the program does collect EHR data if it's consented. So you can do those connections, but trying to figure out the best way of doing research around the return of results is something the program is actively trying to think through. And I think it's all of us, not all of us, know the importance of understanding the impact this may have in the community. And I would love to be able to do that research, but right now, the return information isn't loaded in the workbench just yet. So again, we're building as we go, and I hope we can get there.
My question really was asked and tossed around again, but I just think my question was about the FDA final rule on the laboratory developed tests. So I think that pharmacogenomic tests are definitely relevant for the final rule, which, by the way, is the law of the land. So it's currently, until it's contested or delayed in some way, that's the law of the land. So many of us are looking at that and reading that. It seems that the only exception to an LDT being under regulatory discretion now is that if you are within one health system, right? If you're a lab that's returning only for your patients within one health system, and I think that anything beyond that is going to then be affected by the new final rule of the LDT. So we're reading this and discussing it daily in our lab with our lab people.
The question is for an LDT, do you need to document the clinical utility of what you're doing? And that's where the real cost is. Just saying you have analytical validity is what you're already doing now. That cost is non-trivial.
Even more reason to reach out to your legislators and talk to them about the importance of pharmacogenomics, how it can improve care and reduce spiraling healthcare costs. We need to be having these conversations with the people that represent us.
Should I go? My question was also about the all of us. Is there a component of this study that allows for reinterpretation, going back to the question about that you can't report on novel alleles detected in diverse populations? Is there any way to do that?
Certainly, if there is significant changes in the knowledge base, then we would have to go through the same process of sort of resubmitting what we intend to do. But from change management, we have the ability to recontact and continue the relationships with those participants moving forward. So hasn't happened since from the time of return moving forward. The only time we would pick it up is if during the clinical confirmation something was different, but those are time sequenced to be very, very close. So I can't speak to that. I don't know if it's happened yet.
Thank you.
Other questions? I can ask one then. So Phil, actually building on Laura's first question about equity with the all of us results, and we talked a little bit about this earlier, by requiring that confirmatory testing step, that can pose an equity issue because people with the most education and resources are most likely to get that confirmatory testing. So you also made parallels with direct-to-consumer pharmacogenomic testing that doesn't have a healthcare professional involved, and there are some that now can be used directly in clinical care. Is there a path forward similar for the all of us research results?
If it moved from IDE to a 510K clearance pathway, it could, but that's not the path that was taken originally. So,
and no plans to do so.
Again, I can't speak to the NIH's overall plans on those things, but we chose a particular path, and that's what we have now. 23andMe chose a different path and has different endpoints. Just different paths.
Thank you. And then a question for Christine. What do you think is what's the best way that we can stay up to date with the legislative efforts in pharmacogenomics?
By volunteering, no. I think, again, there's just two key places to go currently. Things do pop up in different states, but the two largest initiatives, American Cancer Society, biomarker, Google it, it'll pull right up, see where it's at in your state. And then for the Right Drug Dose Now Act of 2024, updates are posted at fourthcause.org/RightAct. I just hear peripherally as other things come up. I do know that like NCPDP has a tracking database for all legislation. I'm sure that others exist as well. I don't personally have access to something like that, but I'm sure somebody at an academic medical center probably does and could do a query. But those are the key ones that I'm aware of at this juncture.
Thank you. Do we have another question?
Question for Christine. And I think one of the slides you mentioned there was the testing would cost like somewhere between 8 to 15 cents per patient per month or something like that. Did that also account for money saved from adverse events or that stuff, or is that just for the testing, or how was it's a complex, the whole economic thing is a complex thing. What all went into that calculation, do you know?
Yeah, I'm not sure. I haven't read the entire thing. Those slides were provided to me by ACS CAN, but I can ask for the full report. I am on the PGX task force. But I think it was predominantly focused on cancer biomarker testing. Part of the reason they incorporated pharmacogenomics more specifically into the coverage is because we came to them with predominantly cost-saving data. Although it's not always cost savings, usually cost-effective or cost savings the vast majority of the time, especially as you move to comprehensive panel testing versus drug gene pairs. So that actually really helped, and they found that that information, when they added it to conversations, was helping them advance their agenda when they were talking to legislators.
I will add a little bit, like I think I probably came off negative saying 19 months for things. Remember, this was there was no predicate from their opinion, right? If the folks who are leading research trials start returning results and we all end up going through the same path, your next one through will have a predicate. It'll have this one. If you're not using multiple sites, some of the multiple testing things end up collapsing down. I do believe that 19 months could be a lot more efficient once we have paths moving forward and comparators to go against. So that is not at all, even in, let's say, as we're doing at University of Pittsburgh, we're still pushing for return. I think it's essential that we do that, and I think the path can get faster through the research pipeline. That's obviously separate from what's happening clinically, but I don't want everyone to leave saying don't return results. I actually think it's critical, and I think we can get this to be a lot more efficient and use each other's passes and processes in order to make it more efficient.
The 2C19 variants that you use was essentially any FDA test I've seen is star 2, star 3, and star 17. You have a lot more than that. What did you have to do to get those approved?
The assertions that we were evaluated follows what you would do in a current clinical lab, which was what was evaluated was the phenotype to drug assertions for drug placement. The translations from this star allele to this predicted phenotype, the CPIC translation tables were accepted, and the function was computed from the predicted phenotypes.
That's consistent with us,
yeah.
But at the variant level, there were a lot of variants that we couldn't include because AMP wasn't accepted. Others, we spent a lot of time developing the data around that. I'm wondering if you have data that's available that we could use.
We did not have to submit data assessing clinical validity around alleles in the IDE path. We followed that path through and had to make the assertions at the end, but as for the function assertions and how they translated into predicted phenotypes, we submitted the we submitted the CPIC translation tables and the Forwar allele definitions, and those were accepted in this path. So I suspect if we feel like there's differences, it may be due to the different paths. But all I can speak is to that experience in 2019 to 2020 when we went through it.
I have a question. So Stripe was formed a few years ago to be an avenue for the pharmacogenomics community to interact with the FDA around a lot of these regulatory issues. And it was intended to be part of their collaborative community effort. For all three of you, to what extent has that been collaborative and in the sense of there actually being conversations occurring back and forth productively to kind of move this forward? And in your experiences in interacting with the FDA, to what extent is there appetite for that from the community?
FDA has always participated in CPIC, and now they are in Stripe. I don't know of any instance where we've actually seen feedback from them.
Yep. I'm trying to recall ever a presentation from someone from the FDA that didn't start with, "I'm not speaking for the FDA." Then what's the point? How do we talk to someone that, it has been very frustrating. And this is not a new conversation. I got in trouble, I think it was 2012, speaking at South by Southwest. They're like, Genelex COO calls out the FDA as being too slow to regulate LDTs. great, just latch onto that one. But this isn't it. This isn't new. I am concerned that there seems to be a target on pharmacogenomics a lot. And I don't know how far people go back in pharmacogenomics in the room, but pharmacogenetic testing was covered very well in the early 2000s through, I think, about 2015. There were stacked codes at the time. So genetic testing was covered well. They were like, you're doing stuff to genetic DNA. Yay, good job. Here's some money. But then there were some bad players that came into the industry, and they milked Medicare for hundreds of millions of dollars. And then they decided to throw the baby out with the bathwater, and they shut down reimbursement. LCDs were issued that rolled across the United States that literally closed reimbursement down overnight. And there were a lot of studies that were starting to come out at the time. I co-authored some: dramatic reductions in emergency room visits, hospitalizations, healthcare costs, even death. Who'd like to avoid that? But it nonetheless, it set everybody back. But I will say part of what we can do in Stripe, we've had some talks with the genetic testing registry about getting a kind of industry stamp of approval for laboratories. They're at least covering AMP Tier 1. They're calling phenotypes according to CPIC guidance when it's there. They're making results available in a discrete format that meets XYZ standards. I don't know what those are. We are having a consensus development conference, Stripe, at the end of October, just outside of DC. But we need to, as an industry, make that happen, or we're going to relive what I do not want to relive again that happened in 2015.
I'll just add, if you're going in through the process that exists, like again for the IDE, there are mandated times of responses. I mean, it's 30 days once it goes in, right? So when we've had to do updates or any type of changes, there's plenty of engagement, there's plenty of back and forth, and it gets across the finish line in the time that's required by law. So I think where it's hard is the process is ambiguous. And when the process is ambiguous, it's hard to engage in whatever is likely to be successful and know what you're supposed to do. So I think in that way, it's a little easier for research. If you know you get the distinction from your IRB or the study sponsor that says it has to go through an IDE, there's time, there's money, there's considerations, but that process might actually be more streamlined than what's happening clinically right now.
I did engage with the FDA around clinical decision support tools. They are pretty friendly in chatting with you, which is why we chose not to take raw data files and convert them to diplotypes and phenotypes, but rather start after the lab had issued a lab report, called that themselves and signed off on it, and then start the clinical decision support. You can generate printed reports. You can have access to clinical decision support that always has the latest guidance with links to the references. Make sure it's only provider facing. If you give it to a patient, it suddenly becomes a medical device. But they were more than willing to have a conversation. Kind of, here's what we're doing, here's our references, here's how people access it. It's just for healthcare providers. And they're like, yeah, we think you're okay. They can always change their mind. There's gray areas and such, but they are willing to have conversations.
I think we have time for one more question.
Hi, Christine. I just wanted to follow up on the last thing you said when you said that the reports with recommendations and drugs are not to be patient-facing. Is that correct?
So clinical decision support tools, if they are provider-facing and you enable a provider to make independent clinical decisions for anything you're providing to them. So in UScript, we have summary effects and management and then detailed effects and management with embedded references to every single one of the 19,000 publications we've curated. So you can make your own clinical decision. We print that out as a supplement to a lab report. So lab report, star one, normal metabolizer, Great. Here's a supplementary report that has specific drug and dose change guidance for this patient and the medications they're on, maybe a high evidence drug gene interaction table. These are some drugs, avoid. There's dosing guidance, go, to the clinical decision supporter, we're not worried about it, that have genetic interactions. That can be printed as a supplement, but like lab report, clinical decision support report, that drives people to a dynamic tool. Like everybody keeps saying it, it changes, it changes, it changes. We need to get away from PDFs. We need to get away from paper and start driving people to integrated clinical decision support that is updated as quickly as the evidence evolves.
Thank you. Let's give our panelists a round of applause for an excellent discussion.